Hello, everyone, and welcome to today's live broadcast, Integrating Hematology and Coagulation Results to Enhance Diagnosis, presented by Dr. Larry Smith, Manager, Medical and Scientific Affairs, Hematology, Abbott Diagnostics. I am Alexis Krause of Laboratory, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Laboratory and sponsored by Abbott Diagnostics. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab button found at the top right corner of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Larry Smith. Dr. Larry Smith received his doctorate in biological sciences from Fordham University, Bronx, New York, and completed his certificate in molecular laboratory diagnostics at Michigan State University. He completed his postdoctoral studies in gene therapy and experimental hematopoiesis at Columbia University College of Physicians and Surgeons, New York, and was a research fellow in immunology at Fordham University. In addition, Dr. Smith has a Master of Science in Medical Biology and a Bachelor of Science in French and Biology. He holds multiple board certificates and has published numerous peer-reviewed articles, abstracts, reviews, and monographs. His research interests include laboratory test development for hemostasis and coagulation, HPC therapeutic applications, and flow cytometry. For a complete biography on Dr. Smith, please click on his photo in the top left corner of your screen. Welcome, Dr. Smith. You may now begin your presentation.
Hello, I'd like to welcome everyone to today's lecture on integrating hematology and coagulation results to enhance diagnosis. This program is sponsored by and on behalf of Abbott Diagnostics, and the content of this presentation is consistent with all applicable FDA requirements. Hematology and coagulation testing represent a large majority of tests that are frequently performed in the core laboratory. Now, in some laboratories, the hematology lab and the coagulation lab are separate, and in some cases where the two labs are in the same physical location, we don't always integrate the results from both areas when we're analyzing very complex cases. So what I'd like to show you today is how we can improve management of patients with hematologic disorders through integrating results from hematology and coagulation. And I'd like to do this by describing three disease states that are better managed by the combination of coagulation and hematology results. So I'm going to present three cases. One will be acute promyelocytic leukemia, one chronic lymphocytic leukemia, and one on thrombotic thrombocytopenic purpura. By way of introduction, hematology and coagulation laboratories perform a wide variety of specialized and routine tests for screening, diagnosis, treatment, and prognosis of hematologic disorders. And very often, many of the abnormalities in hematology testing can have a significant impact on coagulation testing. And what we find is that when physicians actually combine the results from both of these areas, it can actually improve and aid physicians in making more rapid diagnoses and even initiate therapies much sooner in their patients. So let's start with a case of a patient who presents with a presumptive diagnosis of acute promyelocytic leukemia and a concomitant disseminated intravascular coagulopathy, or DIC. APL was actually described in 1957 in a patient who presented with a severe bleeding tendencies, and these were fibrinolytic in nature, and an increase in promyelocytes in both the peripheral blood smear and in the bone marrow. This patient also had a rapidly deteriorating course. This was actually described by Dr. Leif Hillestad in 1957. And this disease is characterized by a balanced reciprocal translocation between chromosomes 15 and 17. It's also characterized by the World Health Organization as AML, or acute myelogenous leukemia, with recurrent genetic abnormalities. And that would be a translocation between chromosomes 15 and 17. And this involves a gene called the retinoic acid receptor gene that gets juxtaposed next to the PML gene. Now, under the old French-American British classification, APL is actually called M3. In these patients, we see an increase in promyelocytes and maybe some blasts on the peripheral blood smear, and there's this rapid fatal course with a high incidence of early hemorrhagic bleeding. In terms of some of the frequencies of APL, it's relatively rare, and it comprises about 7 to 8% of adult AML cases. It's usually seen in middle-aged individuals with a median age of 47, and it occurs very rarely before the age of 20. The incidence, again, is slightly higher in males than in females. Turning to the pathogenesis of APL, the defining abnormality is the translocation between chromosome 15 and 17 that involves the two genes that I mentioned earlier, the PML gene as well as the retinoic acid receptor gene. In normal individuals, the PML gene is a gene that plays a role in cell growth and division and even in apoptosis. The retinoic acid receptor gene actually encodes a nuclear hormone receptor transcription factor that promotes the expression of various genes after binding to retinoic acid. So when the actual translocation occurs, what we see is that the PML gene here located on the long arm of chromosome 15 and the retinoic acid receptor gene located here on the long arm of chromosome 17, actually the two genes get juxtaposed next to each other. So this then results in the formation of a fusion protein or chimeric protein, and this protein actually results in the deactivation, or it leads to a transcriptional repression and arrest of differentiation of the promyelocytes. So the promyelocytes get stuck in the promyelocyte stage and they don't continue on in their differentiation pathway. Now, the diagnosis of APL is time critical. So this is because hemorrhage is a major cause of death in these patients. These patients do present with DIC most often, and the DIC in these patients is caused directly and indirectly by the abnormal promyelocytes. If you look at the diagram on the right, here's an abnormal APL cell, and located on the surface are a number of molecules of tissue factor. So there's an overexpression of tissue factor on the surfaces of these cells. This tissue factor is capable of activating the coagulation cascade, and this would lead to a tissue factor-induced disseminated intravascular coagulopathy. At the same time, looking at the image at the bottom of the screen, again, we have an abnormal APL cell, and located on the surface is an increase in annexin 2. And annexin 2 has been described as being an activator of fibrinolysis. So in these patients, we have DIC that comes from an increase in tissue factor, and we also have activation of the fibrinolytic system that would cause for an increase in bleeding as well. So as a life-threatening coagulopathy, it would require prompt diagnosis and recognition of this coagulation defect. So patients with APL, when they present with this coagulopathy, this should always be treated as a medical emergency. Now, in addition to this abnormality within the coagulation cascade itself, the abnormal promyelocytes actually invade the bone marrow, and this then prevents the megakaryocytes from releasing platelets. So we have this compounded condition of activation of the coagulation cascade with a failure to produce platelets. So this all of these combined, we have an increased risk for bleeding in these patients. Now, some APL patients, there has been a description of thrombotic manifestation that can occur with APL as well. So what's the typical flow chart when a patient comes in for testing for APL? Well, it starts with a presumptive diagnosis that's based on a clinical presentation and patient history. This would then be followed by a CBC and differential and routine coagulation screening. The information that we get just from these two types of assays would be enough to confirm the presumptive diagnosis. Later, blood will be sent on these patients for confirmatory diagnosis, and this would include a bone marrow biopsy, fluorescent in situ hybridization, conventional karyotyping, as well as flow cytometry. And then once these patients are treated, we can continuously monitor these patients using the CBC and differential along with routine coagulation screening. So what would some of the results look like in the hematology lab in a patient who presents with APL? Well, if you look at the table that we have here, we notice that we have an elevated WBC count, and this would be consistent with a patient who presents with an undiagnosed APL. In addition, we know that many of these patients who present with leukemia have anemia, and so here we would notice that the RBC count, the hemoglobin, and the hematocrit are all decreased, and this would be consistent with the picture of anemia. And then another result that's very important in the hematology lab would be the platelet count. And here we see the platelet count is decreased, and this too would be consistent with bleeding. Now, if we look toward the middle of the slide here, we see an image of two different histograms, and these histograms will vary based on the type of hematology analyzer that you use. But what you would expect from such a histogram would be at least several populations, lymphocytes, monocytes, and granulocytes. And we would also see a population of abnormal cells that would be present as well. Again, this will differ based on the types of analyzers you have, but it would be a signal to trigger to let the operator know that there is an abnormality in addition to the abnormal numbers that we see. In addition, there are a number of flags that may be generated on our hematology analyzers, and these flags differ based on the analyzer, but one could expect to see a blast flag or an immature cell flag. One might expect to see some kind of flag that indicates immature granulocytes, and even a flag that might indicate a left shift. Again, these will vary from instrument to instrument, but some type of flag would be there to trigger the operator to maybe request a review smear. And in this case, a review smear was done, and what you can see on the right side of the smear are two cells. These are abnormal promyelocytes, and these cells are very characteristic of what was on the actual smear. This actually represented the majority of the cells that were on the smear. What you notice when you look at these cells is that there's a high NC ratio, presence of nucleoli present within the cells. There's an abundance of granules that are located in the cytoplasm. The nucleus has a fine chromatin pattern, and often many of these larger granules in the cytoplasm may coalesce to form aurorods. A manual count was done, and in this particular patient, there were 85% promyelocytes, 1% blasts, and then 9% lymphocytes, 3% monos, and 2% eosinophils. What would you expect in the coagulation laboratory? Well, looking at this table, we have some screening tests, the PT, the APTT, the fibrinogen, and the D-dimer, and what you'll notice is that the PT is elevated, the APTT is elevated, the D-dimer is elevated, and the fibrinogen is decreased. Now, when you take all of these numbers, all of these results, when you take these into consideration, this would appear to be some type of consumption that's occurring, and this would be consistent with DIC. We can also go back to the previous slide and note that the platelet count was decreased as well, and all of this would fit the picture. When you see results like this, it is necessary to differentiate between DIC versus TTP and versus liver disease. And I'll show you a slide later on to show you exactly how we can do that. When we are testing patients for DIC, there are two types of tests that really need to be done. The first is in the coagulation laboratory. The first is a screening assay, and the screening assay, like the PT or the APTT, would simply show you that if the results are elevated, would simply show you that consumption is occurring. We also need a test that would demonstrate thrombin generation along with fibrin degradation, and this is where the elevated D-dimer would come into play. So we've met both of the requirements for identifying DIC in the coagulation laboratory. So as you can see, integrating the results from the hematology and coagulation laboratories provides diagnosis within minutes, and it improves patient management. And so we have a little graphic here just to show you that the test that we do in hematology would consist of the WBC count, and we'd also visualize the increased promyelocytes. We also have thrombocytopenia from our platelet count, and then we have anemia from our red cell and hemoglobin hematocrit parameters. In addition, we also see the coagulation. We would have our elevated D-dimer, elevated PT and APTT. Those would show us that consumption is going on with DIC, and we have our hypofibrinogenemia with our low fibrinogen result in this patient. Additionally, for confirming the diagnosis of APL, certainly one would send a bone marrow aspirate to the lab along with flow cytometry. And as you can see from this particular image on the left, this bone marrow smear is full of promyelocytes. So this would be consistent with the diagnosis. And when we look at the flow cytometry, we have CD13, CD33, CD117, and myeloperoxidase are all positive. And in patients with APL, CD34 is usually negative, along with HLA-DR. So this would then be further confirmation that we're dealing with a case of APL. Additional risk factors can be seen in patients with APL that require monitoring, and these would include things like the cytokine release syndrome, and this is a syndrome that occurs in patients who receive differentiation therapy. This causes these promyelocytes to release a lot of cytokines, and these cytokines have been associated with activating the coagulation cascade. We can monitor therapy in the cytokine release syndrome through various chemical tests such as the potassium, phosphate, calcium, and uric acid. Another risk factor would be the hyperleukocytosis, and of course we would use the CBC and differential to monitor how well we are improving this hyperleukocytosis. And then finally, bleeding, as we said, was a real problem in APL, and often this bleeding can be intracranial bleeding or even bleeding into the lungs, and certainly the routine coagulation test would help us in terms of monitoring the progress in these patients. So the integration of these results contributes to the improved monitoring of these patients, and it allows physicians to treat them more effectively. So let's now turn and look at another case. This would be another leukemia. This would be chronic lymphocytic leukemia. So chronic lymphocytic leukemia is actually an acquired monoclonal disorder that's characterized by a progressive accumulation of functionally incompetent lymphocytes. It's considered the most common form of leukemia in adults in Western countries, and it's extremely rare in Asian countries. When we look at the incidence of CLL, the incidence is slightly higher in males than in females. The ratio is 1.7 males to 1 female, and the disease primarily affects the elderly. The median age is around 72 years of age. There are some cases of familial CLL, and here the median age is a little bit lower at 58 years of age, but these cases are somewhat rare, but they do exist. And then finally, patients who do develop CLL actually have a fairly long lifespan. These patients can live anywhere from 5 to 10 years after diagnosis. There are some reported cases of patients who may die within 2 to 3 years, but this is always due to complications that develop within CLL, and so these cases are not really as common as the cases of CLL, where patients actually live a much longer time frame. Some additional characteristics, if we look at the pathophysiology of CLL, we find that most of the cells are clonal B cells, and again, these cells are arrested in their B cell differentiation pathway. All of these cells have a B cell surface antigen. These antigens range from CD19, CD20, CD1, and CD23 in normal individuals, but in CLL, the CD20 is dim. The other markers are still there, but the CD20 is dim. Now, these patients also express an aberrant T cell marker, and this would be CD5. So we're going to get a B cell marker and a T cell marker on the very same cell. So this would be an aberrant co-expression. When we look at the clinical diagnosis that's required for CLL, of course, 5,000 B lymphocytes per microliter, and this has to be over a prolonged period of at least 3 months duration. There are a number of genetic abnormalities that are associated with CLL. The most common genetic abnormality is a deletion in chromosome 13, and anywhere from 50 to 60% of the cases will have this particular deletion. These are the patients who actually have the longer lifespan once they've been diagnosed. There are some additional mutations that can occur, and when patients present with these mutations, the disease is much harder to manage. So what would a patient with CLL look like in the laboratory? And so this slide shows you 2 different patients. In the very top box, we have one patient, and in the bottom box, we have a different patient. And so what we would see in the hematology lab, of course, would be an elevated white cell count. One of these cases, you see that is 66,000. In the second case, it's 71,000. Again, these patients will present with anemia, and so you can see in both of these cases, there's a slight decrease in the RBC count, the hemoglobin, and the hematocrit, which would be consistent with that anemia. Now, in CLL, the platelet count can be variable, and in both of these cases, there is a normal platelet count, 179,000 versus 222,000. Looking toward the middle of the slide, we see an example of a histogram. Now, these histograms, again, will vary depending on the analyzer that's being used in the laboratory, but we would certainly see our cell clusters with our lymphocyte population, a monocyte population, and a granulocyte population. But what you'll notice is that since we have an increase in lymphocytes in this particular cluster, you can see that it's quite long. It encompasses a much larger area, and this would represent the increased number of absolute lymphocytes. If you're looking at the diagram at the bottom at the lymphocyte population, you can see that instead of being really tight, it's a little bit spread out, or a little spread out going from left to right. And this would simply reflect the variable sizes of the lymphocytes within those populations. So what we'll see in these patients will be an elevated WBC, an increase in the absolute lymphocyte count. Depending on the analyzer that you have, there will be some type of morphological flag. This can range from a blast flag, a variant lymphocyte flag, and other flags based on the analyzer that you have to recognize the population. And often these patients will have on the peripheral blood smear smudge cells. And on some analyzers, these smudge cells may have an increase in fluorescence. Not every analyzer will have these, but we can look for an increase in fluorescence and also identify some of these smudge cells. This is a peripheral blood smear from a patient with CLL, and what you'll notice on the right is these mature appearing lymphocytes. They're immuno-incompetent. It's somewhat of a monotonous picture, whereby they all look about the same. And if you look at the little insert to the further right, you'll see the presence of 2 smudge cells. So these would be those fragile lymphocytes that we were talking about on the previous slide that might have an increase in fluorescence on some analyzers, but certainly we would see these on a peripheral blood smear as a result of making the smear manually, since the cells are fragile. Looking at these lymphocytes, we notice that the chromatin pattern is somewhat chunky. It's been described as having a soccer ball pattern or even a leopard skin pattern. Again, numerous smudge cells when we make a peripheral blood smear, and the RBC and platelet morphology on this particular slide is normal, and that's really consistent with CLL. A confirmation of CLL certainly can be made through flow cytometry. Again, you remember that the surface antigens on normal B cells consist of CD19, 20, 21, and 23, and in CLL, the CD20 is dim. You'll also remember that I said there was an aberrant expression of a B cell marker and a T cell marker. So looking at the histogram on the left right here, we see with CD19 on the y-axis and CD5 on the x-axis, we see a population of cells that would express both CD19 as well as CD5. So this would be characteristic of CLL. In addition, these individuals will express either kappa light chain or lambda light chain expression as well. A number of mechanisms have been described to explain bleeding in patients with CLL, and so some of these mechanisms include things like thrombocytopenia, impaired platelet function defects, acquired von Willebrand syndrome. Some of these patients develop inhibitors to various factors in the coagulation cascade, like factor 8 or factor 9. And there are also some therapy-related mechanisms that cause bleeding in these patients. One of these particular therapy-related bleeding abnormalities that I'll talk about deals with patients who take a drug called Ibrutinib. Ibrutinib has been described as a transformative therapy for patients with high risk and relapsed refractory CLL. And taking the drug by itself has a number of risk factors, such as infections that have been described, bacterial, viral, and fungal. Cytopenias have been described in some of these patients as well. When cytopenias do present in these patients, certainly we can perform a CBC to identify which lineage is decreased, and we can also get a white cell count along with a platelet count for the thrombocytopenia, and we can define the parameters for the anemia. Additionally, bleeding is associated with this particular drug, and many of these patients will present with low-grade ecchymoses and petechiae, and this actually occurs in about 50% of these patients. Major hemorrhage has been reported in anywhere from 1 to 9%, and about 6% of those with fatal bleeding. It's also recommended that when patients are taking this particular drug, and they're also taking an antiplatelet or an anticoagulant therapy, that they be monitored for the risk of bleeding. And certainly the coagulation laboratory comes into play in this regard. The actual mechanism that's associated with this particular drug in patients with CLL is that this drug has been shown to inhibit a number of kinases that are present within the platelet. So if you think about the glycoprotein 6 receptor on platelets will receive a signal. This signal will be generated through the platelet and ultimately result in platelet aggregation or platelet adhesion. And it uses these kinases as it's being generated. It turns out that Ibrutinib actually inhibits these kinases such that the signal can no longer be generated, and this would affect the ability of these platelets to aggregate as well as to adhere. Now, we can also monitor the risk factors in CLL through the integration of hematology and the coagulation lab. And so this slide simply sums up some of the things that we can look for in the hematology lab. Of course, the CBC to monitor the white cell count as well as the anemia, the differential to look for an abnormality within those lymphocytes, or to see if the actual absolute number of lymphocytes is being reduced, and then also the platelet count, and this also plays a part in the risk for bleeding. In the coagulation laboratory, certainly the routine coagulation screen that we've described in terms of the PT and the APTT, and then some more specialized tests such as testing for von Willebrand factor activity and antigen for those patients who might develop an acquired antibody, factor VIII levels for patients who might develop an acquired hemophilia, and then we can do platelet function testing through measuring the closure time as well as platelet aggregometry. So now turning to one final disorder that requires immediate attention, and this disorder is called thrombotic thrombocytopenic purpura. So as we move to the thrombotic microangiopathies, these actually consist of two different subtypes, thrombotic thrombocytopenic purpura, or TTP, and hemolytic uremic syndrome, or HUS. Both of these conditions are really very, very similar, so it will become very important to differentiate between the two of these. The characteristics that we see in patients who present with these two syndromes would be a microangiopathic hemolytic anemia, thrombocytopenia, and microvascular thrombosis. As a result of clot formation inside of these small capillaries, these patients will ultimately suffer end organ damage. In the case that we presented, you saw that the patient had renal failure and she had damage into the heart and lungs. So a number of organs can be affected with end organ damage. TTP is a rare disorder that's characterized by microangiopathic hemolytic anemia. Again, these patients will present with moderate to severe thrombocytopenia, the presence of microscopic clots in the small blood vessels, and it affects about 4 to 6 people out of 1 million individuals per year. Here we have a timeline to show you some of the discoveries with TTP. It was first described in 1925 by Dr. Eli Moskowitz, who described a 16-year-old female who died from a previously undescribed illness that was characterized by microangiopathic hemolytic anemia. She presented with petechiae, hematopoiesis, and fever as well. On post-mortem exam, it was shown that she had numerous hyaline thrombi that were present in her arterioles and capillaries, and these were present in the heart and kidneys. About 11 years later, Bayer and co-workers described the hyaline thrombi and suggested that it consisted of agglutinated platelets. And then 11 years later, Singer and co-workers suggested the term thrombotic thrombocytopenic purpura. So TTP, let's look at the pathogenesis of it just a little bit. We know that we can have what's called a congenital TTP. This is also called the Upshaw-Schulman syndrome, and this is characterized by repeated episodes of thrombocytopenia and microangiopathic hemolytic anemia that begins during childhood. TTP can also be acquired, and when it's acquired, this is generally due to antibodies that form against a cleaving protease. This cleaving protease I'll describe to you is actually called the von Willebrand factor cleaving protease, or ADAM-TS13, a disintegrin and metalloprotease with thrombospondin type 13 motif. It was actually described by Moak in 1982, and he actually described these unusually large von Willebrand factor multimers in these patients with TTP. And when these multimers were released, he proposed that there was the absence or a lack of a cleaving protease that would chop these multimers up. It turns out that these unusually large von Willebrand factor multimers have an increased affinity for binding to platelets. So if you look at the diagram on the right, you'll see a cell here. This is the cell surface with this structure sitting on the cell surface that's called ADAMTS13, and it looks like a pair of scissors. So as the unusually large multimers of von Willebrand factor are being released, this structure, or this cleaving protease, chops the multimers into much smaller pieces, so they lose that strong binding affinity for platelets. Looking at the diagram on the far right, we see this is a patient who has TTP. And what we see here is that the ADAMTS structure here is defective. So we don't have the scissor mechanism that would cut these unusually large multimers into smaller pieces. So we end up with these large multimers that have an increased binding affinity for platelets, leading to thrombosis. Now, if it's an acquired TTP, an antibody would interfere with the action of that particular structure. What do these patients look like in the laboratory when they come in the hematology laboratory? The white count, it can vary from normal to abnormal. In this case, the white count is normal, but we have a decrease in the red count, the hemoglobin, and the hematocrit. So that would be consistent with the anemia, and we have thrombocytopenia when we look at the platelet count. In addition, if we look at a peripheral blood smear and looking at the image in the middle of the slide, we see the presence of schistocytes, these fragile RBCs, and these can range anywhere from moderate to severe in patients with TTP. If we look at the histogram that I have displayed here, and these histograms will vary depending on the analyzer that's used, so they won't all be the same, but this histogram is a little special because what it shows you is a red cell population, and the cells that are falling out of this cluster would be your fragile or fragmented RBCs or schistocytes. And the region down at the very bottom represents platelets. Now, schistocytes often present a problem in the hematology laboratory, and this is especially so if we are counting platelets by an impedance methodology. In this particular histogram, this was counted using optical technology. And what it does is it shows that there is a clear separation between the platelets and the schistocytes. So what this would suggest is that when we're counting the platelets, there would be no interference from those fragmented RBCs. So we would get a much more accurate platelet count using optical methodology. Now, TTP can occur as a congenital disorder, as I mentioned, and HUS is generally an acquired disorder. And on this slide, what we have is another TMA that's similar to TTP, and that would be the HUS. In addition, I have a column here of DIC because we're going to see that some of our results may look similar between all three of these. So some of the differences between TTP and HUS would be that TTP is generally a congenital or can be acquired. It requires very prompt treatment, and this is generally with plasmapheresis, which replaces the ADAMTS13 enzyme. And in terms of its prognosis, in the early days without treatment, it was about 90% fatal. But now with the advent of plasmapheresis, et cetera, this number has been greatly reduced. The chance of TTP reoccurring, especially if it's a congenital form of TTP, the recurrence is likely. When we look at HUS, HUS is related to a bacterial toxin. This is a Shiga toxin or a Shiga-like toxin. So HUS is really an acquired phenomenon. And the bacteria that's been associated with HUS is E. coli O175H7. And this bacteria produces a Shiga toxin that causes this damage. That's similar to what we would see in patients with TTP. In addition, Shigella dysenteriae has been associated with this condition as well. Now, in terms of treatment, rather than plasmapheresis that we would use in TTP, in HUS, we would treat the underlying illness. So we would get rid of the bacterial infection. And in terms of its prognosis, recurrence is really unlikely. DIC can present somewhat of a similar picture with the thrombocytopenia and maybe even some schistocytes, etc. But the cause of DIC is variable. DIC is associated with a number of conditions. We treat DIC initially by we can get rid of the DIC by really treating the underlying illness that's causing the DIC, and then the recurrence would be unlikely. So this table represents some of the coagulation findings that help us to distinguish between TTP, HUS, and DIC. In patients with TTP, the PT and APTT are within normal limits. The same is true for patients with hemolytic uremic syndrome, and in patients with DIC, the PT and APTT are elevated. So that's one of the distinguishing characteristics between DIC versus TTP and HUS. With the D-dimer testing in patients with TTP and HUS, the results are normal to slightly elevated, whereas in DIC, the results are generally elevated always. When we look at the fibrinogen assay, the value for the fibrinogen will range from high normal to increased in patients with TTP as well as patients with HUS. However, in DIC, the fibrinogen level is decreased. So this low fibrinogen would help us to differentiate DIC from TTP and HUS. And then finally, in those patients with thrombotic thrombocytopenia, there is a special test that we can do to identify the level of ADAM-TS13 activity. This would be the activity assay. And in these patients, it's less than 10% in most of the cases. Now, there are two additional tests that we can do along with the ADAM-TS13 activity. That would be the inhibitor level as well as the antibody titer. These two additional assays would be reflex testing that would be done, but they would only be indicated in a patient that had an acquired TTP as opposed to a patient with congenital TTP. In the case of HUS, we can also measure the level of ADAM-TS13 activity, and in these patients, the level is much higher than the 10%. The inhibitor and antibody reflex testing would not be indicated in patients with hemolytic uremic syndrome. And then finally, in patients with DIC, these tests would not really be indicated at all, but if they were done, the results would be within normal limits. So in summary, we've reviewed 3 disease states where integrating the results from the Hematology and Coagulation laboratories lead to improved patient management and outcomes. As we can see in the case of APL, the combination of hematology and coagulation results supports early treatment of these patients who present with APL while waiting for more specialized confirmational tests to be performed. As we can see with CLL, the hematology numerical results that we get from our analyzer show evidence of lymphocyte abnormalities that are associated with this condition. In addition, the CBC and coagulation screen can be used to monitor bleeding risk that we see in many of these patients. And these tests are very important when it comes to those patients with a therapy-related bleeding case. And then finally, in those patients with TTP, even though TTP is a rare condition, the presence of these RBC fragments that we see on our peripheral blood smear and the RBC fragment flag that's available on some analyzers may provide an indication of the presence of TTP in these patients. In addition to looking for these fragmented red cells, we know that routine coagulation tests that are combined with an ADAM-TS13 evaluation also provides critical differential diagnosis between TTP, HUS, and DIC. So with that, I'll open the floor for questions.
Thank you, Dr. Smith, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, are there ways to improve the sharing of results between coagulation and hematology when the two labs are not physically joined together?
Thank you for that question. There are ways that you can improve sharing the results when the two laboratories are not physically located, but it would probably require some type of middleware such that the people on the hematology side would be able to see the results simultaneously from the coagulation side and vice versa. That would probably be the best way to see the results. Of course, you could see the results in the LIS before you actually accepted and sent the results off, but that would probably slow the process down. So I would think that a middleware solution might be a better fit in that scenario.
Our next question is, WBC is very important to monitor therapy in this case. Is WBC count correct?
Thank you for your question. I think that's a very good question and certainly one that we should all look into. I think it may depend on the type of methodology that you use to counter WBCs. Using optical technology, I don't see any problem with the WBC count being correct as long as it's within the AMR for the particular analyzer that's being used. With impedance technology, I'm not really sure, but I can imagine that there might be some possibility of the assay not being totally correct or the WBC count not being totally correct, especially if there's some type of interfering substance that might interfere. This would require that a laboratory would validate the extremely high counts and the counts as they return to normal and make sure that interfering substances do not interfere with the particular methodology that they're using.
Dr. Smith, are there hematology analyzers that can detect the presence of smudge cells?
Thank you for your question. There are some analyzers that may generate a particular signal when they are measuring fluorescence. It turns out that schistocytes are these fragile WBCs. There has been some association, and if an analyzer is capable of showing an increase in fluorescence in a particular cluster population, then this would be possible. I have seen this on some of the optical analyzers.
Our next question, what role does the core lab play in patient follow-up after treatment for any of the diseases mentioned?
The core laboratory plays an extremely important role when it comes to follow-up. If you think about the case of APL and the increased bleeding risk, certainly the coagulation laboratory would be able to monitor the effectiveness of treatment, whereby we would look to see that the values return to the normal limits. By the same token, the hematology lab would also play a very significant role because we could see immediately that our platelet count is returning to normal, and the elevated WBC count would be returning back to within the normal limit. In the case of the CLL, the same would apply. We can use both the hematology and coagulation laboratories to monitor improvements in some of these bleeding abnormalities as well as the anemia that these patients present with.
All right, Dr. Smith, are there specific signs that might help to identify TTP sooner so that the proper laboratory test can be identified sooner that would lead to earlier patient intervention?
Thank you for that question as well. There are some specific things that we can look for. Certainly, the clinical condition that the patient comes in would be very, very important, but in the laboratory itself, the presence of schistocytes is really very, very important, and the sooner that we can identify those schistocytes and report them out, certainly a physician could start treatment at a much sooner time point. So if you have an analyzer that can actually identify these schistocytes, this would be done within a minute of running the sample. This really could help improve the turnaround time when it comes to treating these patients. Otherwise, if you have to depend on a peripheral blood smear, you certainly have to wait until the slide is made and wait until it's stained, and then for someone to look at it. So by having an analyzer that's capable of identifying these fragmented RBCs, that could certainly be very advantageous.
What additional testing would you do in coagulation to identify acquired von Willebrand disease?
To identify acquired von Willebrand disease, we would need to do some more specialized coagulation testing such as factor VIII, von Willebrand factor activity, and von Willebrand factor antigen. And one of the things that we would look for would be a discordance between the levels of von Willebrand factor activity and the antigen. In addition, there are some additional tests we could use, such as multiple analysis, et cetera, but to just get something very quick, we could just simply do a factor VIII, von Willebrand factor activity, and von Willebrand factor antigen. These would be additional tests in addition to the routine coagulation tests that we would normally do.
Dr. Smith, do you need to perform platelet function studies with the closure time and platelet aggregometry simultaneously?
You really don't have to perform both of these assays simultaneously. You may get enough information from the platelet function screen depending on what you're actually looking for. Either you'll get a prolongation in your collagen epinephrine closure time or your collagen ADP closure time. And this may be sufficient as long as you're correlating it with the patient's status, et cetera. Very often, aggregometry may be done, but it would require that it be done right away, and so you really couldn't wait. So if you were looking for something a little more complicated, platelet aggregometry would be your next choice. But I think just a platelet function screen, if it's something very simple that you're looking for, should be sufficient. Again, I would just reemphasize that just because the platelet function screen is normal, there still could be a platelet abnormality that would be identified through platelet aggregometry. So it really depends on the clinical condition that you're looking at and what the patient is actually presenting with.
Is APL the only mutation that can occur in AML? If not, what are some of the other mutations, and would they respond to therapy the same way?
Well, APL is only one mutation that can occur in the subset of AMLs, and I believe that this question may have been more focused on the actual mutation that occurs in APL, the 1517 translocation. This is the most commonly occurring mutation in APL, but there are some other mutations that can occur, and it is important to understand those additional mutations because the treatment would be different. The response that we get in patients with a 1517 translocation and the therapy that we're giving them would differ from the response that we would get if there were a different type of mutation. And the same thing would occur in CLL, as we discussed as well, in the deleted chromosome 13 mutation. If there's a different mutation, these patients may not respond to therapy very well. So confirming the actual mutations in any of these hematologic disorders really is essential when it comes to therapy.
And Dr. Smith, it looks like we have time for one more question. How important is the routine coagulation screening in DIC? Are there other coagulation tests that can be performed in the core lab to diagnose DIC?
The routine coagulation tests are really very simple, very cheap, and they do offer a lot of information concerning the overall picture. The routine assays that we do for patients with DIC, we need to show that there is this consumption that's occurring. So the routine PT and APTT would be prolonged, and that would be suggestive of consumption. The decrease in the fibrinogen would go along with this very same pattern. So those are really very, very good. On the hematology side, the decrease in the platelet count would also be very consistent with DIC. There are no specific tests that can be done that when they're positive, it says I am DIC. So the routine tests are really some of the better ones that we have, but most importantly, it's a combination of these assays that would lead to a diagnosis.
Thank you again, Dr. Smith. Do you have any final comments for our audience?
I would just like to wish everyone a happy laboratory week. I hope that you enjoy many of the festivities that you have planned, and thank you all for listening to this lecture. If you have any questions, please feel free to send an email. We'll try to get back to you as quickly as we can.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today or those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of the registration. We would like to thank LabRoots and our sponsor, Abbott Diagnostics, for underwriting today's educational webcast. This webcast can be viewed on demand through November of 2018. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.